My name is Clay. I am a research technician here in the lab. I have two areas of research. I am currently heading a group of 4 undergraduate students where we work with a neuroscience lab, and we basically look at the effects of chronic opioid use on the gut microbiome, and specifically how that changes enterococci populations in the gut. And then we also have this project that I'm working on now in collaboration with a partner who's a PhD student here, as well as one of our collaborators up at Michigan, their medical school, where we are looking at these pairs of enterococci that were isolated from blood, and we're seeing how they became resistant to the antibiotic depth.
Illustrate what we're seeing in each one of these?
So this is a pair. So this is one that has been isolated from blood that is susceptible to daptomycin, and this is a homologue that is resistant to daptomycin. So we're getting ready to grow these in overnight cultures, and then we're going to isolate them and do lipid extractions, and then send them to another one of our collaborators at Duke University, who their responsibility is basically like lipid identification. So we're going to see how the changes in lipid composition of the cell membrane are that allows them to be resistant to daptomycin.
So I'm Tyler. I'm a senior in my undergrad, and I'm here I'm here for fun, but I'm also here for credit. And for my project, I'm doing a continuation of the project or a series of experiments that we do in a lab course, the microbiology lab. So what I'm doing is I'm looking into soil bacteria. The main reason is a lot of antibiotics that are used clinically come from soil bacteria, and with antibiotic resistance kind of being a problem now, what I'm doing is I'm looking for new soil bacteria essentially and seeing if it can produce antibiotics that might be clinically relevant. So to do that, I'm just plating a lot of soil, and here there's like a bunch of different bacteria, and then what I'm going to eventually do is I'm going to select just whatever bacteria I'm interested in, and one of the tests that I'll do following after is I'm going to grab what are called like tester strains, which are like specific bacteria that we know can potentially like cause issues, and I'm gonna spread it onto a plate. I'm gonna take one of my soil isolates, I'm gonna streak it inside and see if it can produce antibiotics against it, and that should be seen in some like a clear zone around the bacteria.
Very good.
Yeah. A little side thing I'm also doing is I also want to see if I can get bacteria to also produce some pigments, just to see some colorful bacteria. And how I've been doing that is I just left the light on here. Pigment production in bacteria functions as like a barrier to like light, like UV damage kind of stuff like that. So by growing them under the light, I might be able to induce bacteria to grow or to produce pigments as well. So like we can see some colored ones.
I saw a shot of orange.
Yeah, there's a lot of orange. There's like black bacteria, yellow, and then there were some green ones and pink ones.
Now, how do you identify, what is happening?
One of the experiments that we do later on is we're going to do PCR testing for a specific gene called the 16S RNA gene, which is a gene that's typically conserved and unique to bacterial species, or one of the techniques you can use. And then we're going to use that to be able to identify the bacteria.